My name is Britt Nicholson. I'm a Senior Vice President for Development at Mass General and an internist here at the hospital. And on behalf of the institution, I'd like to welcome you to Mass General's COVID-19 Clinical Trials Update. Just a few housekeeping items to begin with. First of all, you do not need a microphone or a video capability to participate in this webinar. And at any point during the webinar, you'll be able to ask questions, and you can do it anonymously if you prefer, through a Q&A feature, which is at the bottom of the screen. One thing I would ask, if you could keep the questions as focused as possible, so we'll have the chance to answer as many as possible. Now, the webinar will be recorded, and a shareable link will be provided after the event. So in addition, a follow-up email will be sent with all questions and answers to today's event, even those questions that we may not be able to get to answer during the live webinar. You know, the current situation at Mass General is that as of 3 weeks ago, we've reached the peak of new COVID admissions, and those numbers have fallen dramatically, as has the number of critically ill COVID patients. This has enabled us, following state guidelines, to begin to resume, albeit on a limited basis, some non-emergent procedures. We're actively preparing for the resumption of routine procedures and routine outpatient visits when given clearance by the state. And that clearance will be based on continuing favorable trends in COVID admissions, COVID deaths, and testing results. But as we remain focused on the care of our patients, we're also recognizing we have to address the financial consequences the pandemic has created for this institution. Now, just as we were prepared to meet the clinical challenges posed by this virus, as an institution, we're also uniquely positioned to immediately address the research questions which need to be answered if we are to create the therapies needed to safely resume our day-to-day lives. We are the largest hospital-based research program in the nation, with more than 8,500 researchers working across more than 30 institutes, centers, and departments. Now I'd like to introduce Dr. Keith Flaherty and Mr. Rick Frisbie, today's speakers. Rick is one of the founders of Battery Ventures, a Boston-based venture capital firm, and Rick is also an honorary director on the MGH President's Council and member of the MGH Fund Leadership Council. Dr. Flaherty is director of COVID-19 clinical research for the Mass General Hospital and also serves as director of the clinical research for the Mass General Cancer Center. He served as principal investigator for numerous first-in-human clinical trials, and his work has directly led to 5 FDA approvals. And so right now, I'd like to hand things off to you, Dr. Flaherty.
Thanks, and thanks, Rick, for co-hosting this discussion. So I thought it might be useful to maybe set the context a little bit. Maybe first, just to explain, as Britt just highlighted, that an oncologist is directing COVID clinical research. What's the logic of that? So if there is a logic, it's that, as many of you are likely aware, cancer therapy development is a very large enterprise. There are many, many therapies being developed simultaneously. And at cancer centers like ours, we are constantly trying to bring our scientific insights to bear in the therapeutic development of new treatments. And we do that for dozens of therapies simultaneously, and thereby simply have just a larger pipeline of therapies in development at any given time in oncology compared to any other therapeutic area. In addition, we're always trying to understand the biology of the disease that we're trying to treat, as well as how it reacts to and responds to the treatments, the new treatments, the investigational treatments. So we do sort of parallel research, if you will, therapeutics and biology in the patients. And that was exactly the challenge that we needed to stand up about 3 months ago when the wave of COVID-19 was coming to Boston. We needed to essentially instantaneously capture the talent, which is extremely broad. As Britt alluded to, we have a very large research universe, really, and that's very basic laboratory scientists, translational scientists, and clinical investigators. And I would say that, you know, roughly 200 of those individuals were immediately full-time COVID-19 researchers, enlisting themselves and their groups. So here I'm speaking to the sort of lead investigators, several of whom have you know, large laboratories and so on. And coordinating that effort is not something that had happened before for reasons that you're probably aware of. And let me just kind of briefly account for the sort of the way in which COVID affects the population, and how we've reacted to that. As many of you know, COVID is a coronavirus, which is amongst a family of viruses that actually cause common colds. But this is a virus that is not only more virulent in terms of its ability to spread quite efficiently, but even more striking is its ability to cause severe illness. And that illness, essentially like all viral infections, is not a consequence of the virus itself. In other words, it is not the virus itself directly damaging tissue and causing life-threatening complications. Rather, it's the response to the virus. So it's the inflammatory response to the virus that is so robust. So this is true with influenza, and it's true with COVID-19 or SARS-CoV-2. So knowing that history of viral inflammatory responses, we immediately enlisted immunology, inflammation experts. Of course, we needed pulmonary critical care experts. Of course, we needed infectious disease experts, virologists, and teams of them, really, to try to understand the basis of why this virus causes disease so differently than other ones. But think of this as a pyramid, where at the base of the pyramid, we have a very large number of people in the population who are infected but are asymptomatic throughout their entire disease course. And as many of you know, that's typically younger people. So children, young adults, even up through the Middle Ages. It's very, very likely that people who contract this infection will in fact be asymptomatic through their course. And we've actually done some essentially overnight epidemiologic studies in our Boston community to document how many asymptomatic cases there can be, even in a hotbed of symptomatic cases. So the ratio we think is actually far greater, of cases, asymptomatic cases to symptomatic cases. There are those who are mildly affected, there are those who are moderately affected, and then of course there are those who are severely affected and who are hospitalized. But ultimately, as we have tried to address the problem in the hospital, understanding this deleterious immune response, this overly exuberant immune response or inflammatory response that causes tissue injury and life-threatening complications, we've had our eye towards moving toward where the problem starts, in the community amongst the mildly affected population, and even those who are asymptomatic. As we'll discuss, ultimately we want a vaccine. With any viral infection, we know that can be ultimately the way out of this whole scenario, and we wish we could accelerate those timelines, and we will absolutely roll up our sleeves and contribute to that effort in terms of accelerating the clinical validation and ultimately adoption of vaccines. But along the way, we have a problem in front of us in terms of trying to disrupt this overly exuberant inflammatory response, which has clotting complications and other features that I'm sure you've all read about, and then ultimately develop direct antiviral therapy that can be deployed not only in the hospital population, but broadly in the outpatient population. It's in those individuals where we think we probably don't need to try to disrupt the inflammatory consequences of the disease or the thrombotic complications and those kinds of severe issues, rather direct antiviral therapy. And we'll talk about that in a moment in some detail. So think of this as, yes, a viral infection analogous to influenza, but with sufficient novelty in the human population that many individuals, we think because of really genetic underpinnings, I mean, in germline or inborn variation amongst us, that has some people's immune system responding so much more vigorously than they would to any other viral infection, including influenza. And that's what's led to this higher rate of hospitalization and unfortunately even fatality compared to those other types of infections. So the scale of the problem you're aware of, I just wanted to sort of shape, if you will, the nature of it and the way in which it manifests. We, of course, were trying to instantaneously respond to the problem as it confronted us with severe patients, presenting to our clinics and to our emergency department requiring respiratory support, even ventilatory support, and come up with therapies, which we've been evaluating quite vigorously over the past now almost 3 months. We've learned a lot about the biology of the disease in the meantime. And now that's really arming us with what we need to know to be able to now deploy a much larger base of clinical investigation in the ambulatory setting, trying to intercept the disease at an earlier point, prevent people from being hospitalized. If we can do that successfully, even in advance of a vaccine, that really takes the teeth out of this pandemic in a way that allows us to think about being able to return to work, yes, with some risk, more risk than just sheltering in place indefinitely. But none of us essentially can tolerate that. And so we need to make advances in real time so that we can actually protect people from stepping back into their regular lives and economic activity and the like. So with that as a foundation, I thought maybe we would transition to Rick's framing of the discussion and try to tackle some questions from there.
Sure, okay. Thanks very much, Keith. I think you explained that, very well. And although I'm familiar with a lot of this stuff, I think I learned a little bit more listening to you today. Anyway, the reason that I'm involved in this call today is because my wife Lisa and I decided, it's probably now about a month and a half or 2 months ago, to make a substantial contribution to the clinical trial activities at MGH, because we felt that was the best possible way for us to have a material impact on the fight against the COVID-19 pandemic. At the heart of our decision was the desire to be part of this effort somehow. And I'm not able to do the research that Keith is doing. And so, we decided to go to our checkbook. And, in doing so, a lot of it was based on, sort of the way I think about things coming from the venture capital world. Where in venture capital, I learned the value of funding projects at an early stage that can be real game changers. In this case, my conclusion was that would most likely be therapeutics and diagnostics that would eventually, lead us to, or eventually we'd have a vaccine, but in the interim, we've got to do something. And we can't close down the country and watch people die, waiting for the vaccines to come along. So we need to do something, to effectively fight this virus that allows us to get back to work and, get people out there doing the things that they, normally do. I think with the right drugs and tests, we'll be able to get, back to the business of our country before this, the economic damage becomes cataclysmic. And of course, before the death toll becomes, something that's really intolerable. So I think it's critical that we get things moving quickly here. And drugs will hopefully be the solution, to buy us the time that we need to get to the vaccines. I'm sure you've all seen, recently Dr. Fauci has been somewhat optimistic about some of the vaccines, that are in process. And we all hope he's correct about that. But we'll still, in the interim, we need something, to get us there. So for me, it was sort of an easy decision. Mass General, which I've been associated with, for a number of years, was, the clear choice for me for a group to work with. I think we all on this call know that it's one of the top healthcare institutions in the world. And in my early discussions with members of the Mass General community, it became apparent to me that Mass General was, sort of in the middle of many of the clinical trials that were either being, done or evaluated. And whether Mass General actually does a clinical trial, that they're still involved in the process of working with other institutions throughout the country and internationally to decide, which are the trials that are most worthy of being done. So Mass General plays a really critical role in this process, determining what gets done and how it gets done and who does it. so far, this effort that Mass General has been involved with, has cost several $1,000,000, for Mass General. And over the course of the next 3 months or 6 months, whatever it takes, it will easily be another $10 million or perhaps more, depending upon how long this all takes. So my wife and I believe that contributing to this effort at MGH, really has the potential to make a dramatically outsized difference in finding a solution, to this problem that exists, throughout the world. I'm hoping that many others will, participate along with us in this funding effort, because it can have such a significant impact on the health and economic well-being of everyone. I believe that this is the first time in my life that I've ever made a financial commitment that was part of something so big and so immediate and so important. And that isn't to say that being involved in cancer research isn't important, because it absolutely is, but it's different than what we're facing here. This is critical in the short term, and we need to do something about it. In my lifetime, there's nothing else that's, even been close to this. So I feel very good about, trying to help this effort in the small way that I can. So that's why I'm here.
So Rick, I thought it might be useful to expand a little bit on these types of therapies, which again, I'm sure everyone on today's call has been reading on the topic of repurposing therapeutics, right? So repurposing antiviral therapies, and repurposing anti-inflammatory therapies, and then a transition to more COVID-19 specific therapies. So maybe let's maybe kind of try to frame that a little bit. There are really 3 direct antiviral drugs, one from HIV, and 2 that were developed originally to try to deal with Ebola virus and MERS, the Middle Eastern respiratory virus, which is actually in the coronavirus family, like SARS-CoV-2 or COVID-19. So really only 3 drugs that really have a direct antiviral mechanism that we inherited, if you will. The HIV drug, ritonavir, unfortunately actually failed in a very early randomized study that was conducted in Far East and Europe before COVID-19 even hit the US. Remdesivir, which I'm sure everyone is aware of now, is one of the other 2 drugs, favipiravir being the third. Remdesivir actually did register a positive effect, and sufficient effect actually warrants emergency use authorization by the FDA. But it was for an in-hospital population, and it's an intravenous drug given twice daily for 5 days, and doesn't really set the stage very well for what I mentioned before, which is this much broader effort that we want to undertake over the coming few months in the ambulatory setting. Be very challenging to administer that therapy in the ambulatory setting to amongst thousands of patients, even, let's say, in the next clinical trial. Favipiravir happens to be an oral drug in the midst of conducting a randomized trial of that as we speak amongst 3 centers in the Bay Area. We lead that consortium, and we're very hopeful. But then we've exhausted the list of direct antiviral drugs. Now, what's fascinating is that there are a group of laboratories, about 8 to 10 of them worldwide, that have been screening existing drugs, FDA-approved drugs for all variety of conditions, to see if they have activity against any of the components of this 15-gene organism, SARS-CoV-2. And it turns out there's about a dozen of those that score in those screens, and repeatedly across multiple laboratories. So we are setting up a clinical trial right now that we refer to as a platform clinical trial that's going to allow us to basically ingest those agents and quickly vet them, essentially simultaneously in parallel. Thankfully, the vast majority of them are oral drugs, so they do potentially lend themselves to the outpatient setting. But we know full well, to your point about kind of an investor mindset, they're not all going to work. Even the vast majority of them aren't going to work. But if we can pick up another win or 2 out of that category, I think that certainly keeps us moving in the direction we want to go. The anti-inflammatory drugs, I'll maybe skip over for the simple reason that it's the most chaotic of the areas. There are so many anti-inflammatory drugs, and there's varying rationale for using them. And yes, we are investigating them for the in-hospital population, but it's really direct antiviral strategies that we need for the broader base of the population. And so I thought it'd be worth putting on people's radar the fact that we have two strategies there, here and now. One of them is convalescent plasma, which again, I'm sure many people have been reading about. This is actually through the generous donation of people who have been affected by and recovered from COVID-19. We have now hundreds of people, survivors of COVID-19 in our region, and that, of course, that number continues to, who have volunteered very generously to donate plasma, which requires essentially a few-hour procedure that's like donating blood, but just a little bit more time in a chair than a blood donation. And then that gives us the material from which we can isolate antibodies and administer those. That's actually an old concept. It's been tried actually for 100 years in various settings, not in very many rigorous clinical trials. But we have orchestrated another Boston area or Bay Area, if you will, consortium evaluating convalescent plasma in a rigorous way to really understand which of those antibody responses that patients mounted are actually able to be transferred successfully. But in parallel with that, we hope, in fact, this week, before this week ends, we will dose the very first patient with one of the prospectively developed neutralizing antibodies that are engineered in a laboratory. In other words, prospectively made for COVID-19. We're very optimistic about this approach. It's actually something that technologically has never been mounted in past epidemics or pandemics, but this simply reflects where we are in the evolution of the biotechnology sector, that so rapidly these therapeutics were able to be developed. There's 3 companies that are essentially co-leading this space. We're working very closely with 2 of them right now, and as I said, hoping to dose the very first patient even this week. Those are purpose-built therapies. We actually are very optimistic that a combination approach, a 2-drug approach, is likely where we need to be, and that might actually be a direct antiviral with one of these antibody type strategies, and that's the type of platform, if you will, we hope to elaborate over just the next month or 2. We have to move in real time. This is the challenge. Companies have been incredibly generous in terms of making their therapies available to us, but organizing our research staff, executing these trials with maximum speed, and simultaneously capturing the types of specimens, blood samples, sputum specimens, to understand which patients are actually benefiting, what's the biologic basis of that. This is what really is the basis for our discussion today, to be doing all of this in a time frame that we've never done before in any aspect of biomedical research. Oncology we think of as actually being quite a fast-moving area. COVID-19 makes oncology look like it moves in geologic time.
Keith, I don't want to put you on the spot here, but what do you think the chances are that someone will come up with a drug or a therapeutic that that will work to some meaningful extent?
Yeah, I'm pretty optimistic that amongst the pool of therapies that we've talked about, just at a high level here, that we're gonna score some more wins. We actually, I would say we see remdesivir, which, by the way, we were the highest accruer to the randomized trial that led to its approval, so we invested quite vigorously in its evaluation. We see that as a building block of sorts. I mean, I mentioned its limitations, its obvious logistic limitations, but it tells us that this virus is not so wildly different from others. Some of the rules that we've learned in terms of how the immune system can respond effectively and mount the type of clearing, viral clearing response, we think will pertain. And so back to that category of repurposed drugs that can be readily repurposed from these laboratory screens, I think it's reasonable to be optimistic because we have benchmarking data with remdesivir in those same screening systems that we're going to score another couple wins out of that category. And this, I think, will play in a way that's similar to HIV, which, just wind back the clock, you were likely all there at the time. The very first drugs, idobudine or AZT, it moved the needle, but very transiently. Right, a single drug approach basically was not enough to keep HIV at bay. 2 drugs, magnificently more effective. 3 drugs ultimately has turned HIV largely into a chronic disease. So I think what you're asking, Rick, is what's going to get us to the equivalent of a 2-drug regimen with HIV? And I'm suggesting I think it's probably going to be a 2-drug regimen for COVID.
Right, which is fine as long as we get there.
Yes, and quickly.
Keith, one of the questions that just came in was that you were, which plays off of the discussion around the true drug therapy. Are there other examples of where a 2-drug therapy has worked, and why does it give you such confidence that this is an area to pursue?
You know, it's really only HIV in the viral space, and that's I would say, well, we've frankly never has there been such motivation to pursue multi-agent, regimens to combat viral disease as there has been now. Seasonal flu, admittedly, is a potential motivator of similar campaigns. You're all aware of Tamiflu, for example, which does have an effect, but has to be administered very early in the course of disease to have any real efficacy. The problem with flu, and it turns out to be a greater problem with flu, oddly, than COVID-19, is that the virus mutates so quickly that it's difficult to come up with a stable portfolio of drugs that will be useful against the unchanging or stable components of the virus. Now, having studied COVID-19 over the past 3 months, we've been sequencing the virus amongst hundreds and hundreds of our patients to map out where mutations occur and where they don't occur. And it appears to not be quite as labile a virus as seasonal flu, which is otherwise the one that causes the most mortality, certainly in the population. So I would say that basically the substrate hasn't been there for seasonal flu. Certainly it was there for HIV. And I would say that really is the precedent that we're hanging our hat on here. I could bore you to tears with the cancer examples of 2-drug regimens, which is what we've been working on and successfully so in several instances in the oncology space for the past decade.
So the another question that just came in has to do with what's the process that you've set up to choose the most promising areas to pursue, and what's the near-term cost of being able to fund these at the MGH?
Yeah. Well, on the process side, look, we've been in the era of team science now for several years, broadly across the institution. And so when it comes to deliberation and strategic planning, that's been a team sport. So literally overnight, now about 2.5 months ago, we pulled together a team of infectious disease specialists, immunology specialists, pulmonary critical care, cardiovascular thrombosis experts, all of the domains where we knew the virus was already exerting some effect or wreaking some havoc. And having scientific leaders in each of those domains on our steering committee, as we call it, and our scientific review committee for trials, it's allowed us to take the 85 trials that have come to our doorstep, some by MGH investigators, some extrinsically from pharmaceutical companies, and some from the consortia, including NIH-funded consortia, that some of our leaders are chairpeople of. And so we have this broad array of bright ideas that have been brought to the doorstep, but we needed to come up with a strategy by which we vet those in a way that really brings our scientific firepower to bear. This is entirely analogous to what we do in the oncology space. So it was another natural fit, if you will, in terms of what my experience has been able to call on. But to this point about how do we ultimately execute in this arena with optimal impact. What I would say is that basically, I'll repeat the point I made before. Never before in modern medical history has the private sector, so small biotechs up to big pharma companies, been so generous in terms of offering access to their therapies. And some of these companies have enough conviction that they're willing to put capital to work as well and are supporting the conduct of these trials. That's the minority, in fact, a relatively small minority. A larger proportion of them say, well, look, we're developing these drugs for other purposes, but we are certainly willing to make them available. And that's a precious resource in its own right. And those are the substrate for what we call investigator-initiated trials. And even if it's an NIH-supported study, it would still fall roughly in that brick where essentially the drug's being made available, and not quite the support needed to actually be able to staff these clinical trials with the research personnel needed. I don't mean physicians, I mean the professional staff who orchestrate the trials, research nurses, research coordinators, and the like. And when Rick was talking about cost before, that really is the cost. It's the human capital in terms of the conduct of these studies. And at the scale that we've been doing them, we've been accruing hundreds and hundreds of patients to these trials. I mean, this has been the most productive single area of clinical investigation on a per month basis in MGH history. There's just never been a time like it. So we've been putting a lot of human capital to work. It's been absolutely inspiring to see it. We need to continue to be disciplined going forward, as I've suggested, bringing our sort of scientific acumen to bear, picking amongst, as I said, 85 clinical trials. There were 15 of those that we thought actually had some scientific merit to be worth the investment of our limited bandwidth. People have to remember, of course, that for the hospital population, there couldn't be a more stressful time in which to try to conduct research. I mean, simply could not be a more stressful time, given the concerns of infection to research staff and so on. But it's been an absolutely inspiring enterprise to be a part of. And the work lies ahead, as I've said, particularly to scale out to the population and to get to the ambulatory setting and get ahead of this disease, which is really at the transition point at which we find ourselves today.
So I think we've got time for one more question before we wrap up. This one came in. Do you think a vaccine will provide seasonal immunity like the influenza typical trivalent dose, or lifetime immunity like polio?
It goes back to that comment I made about the mutation, mutational abilities of the virus. So I suggested that influenza, in fact, is more highly mutable, more changeable, which is why we only get seasonal protection from that virus. I don't mean to suggest that SARS-CoV-2 is incapable of mutating. We've been watching it mutate. We've already documented, again, now almost a few hundred individual mutations from real patients and their sputum samples. So we know it's mutating, but we also know there's domains that won't mutate. And so if we can engineer the vaccine so that it elicits a response to those non-changing domains, that's the hope in terms of being able to generate a vaccine that actually gives us a more permanent protection. I think people need to be prepared for the possibility that there's going to be actually multiple vaccines that will at least partially be effective. And we may need an iteration of them to finally get to the level of protection that we all ultimately desire. We'll take anything that gives us 60, 70% efficacy or greater. That'll start to protect the population on towards something more like 90% plus, which would really tamp down the ability of the virus to maintain itself in the population.
Well, we've got a number of questions that we haven't been able to get to, but Rick, Keith, thanks very much for a stimulating discussion. I'd like to thank all of you for joining us. And Rick, any final comments before we sign off?
Well, I just want to say thank you, to everyone on this call, and to Keith in particular, who I think did a terrific job, talking about where we are and where we need to go with this. And although I've spent a lot of time reading about it and studying about it, I've learned a number of things today just listening to Keith. So, I think it was rewarding.
Well, Rick, we couldn't be more grateful for your leadership in this extremely trying time. Thank you for your past leadership and Tide at Mass General. But as I said, we've needed to respond to this problem in real time and take this country's largest research community and bring it to bear in the most impactful way. So we're extremely grateful for your leadership and support, and thank you all for participating and listening into today's discussion. I'd be more than happy to follow up, with individuals as well to talk further about our efforts.
So on behalf of the institution, again, thank you for attending, and please be safe.